Hello, everyone. My name is Shefali Setia Verma, and thank you to LabRoots for giving me the opportunity to talk today about this topic, bridging women's health and genomics and electronic health records, biobanks. Just quickly, something about me. As I said, my name is Shefali Verma, and I'm an assistant professor in the Department of Pathology and Lab Medicine at University of Pennsylvania. Background, I have gotten a Ph.D. in bioinformatics and genomics, and right now my lab focuses on linking electronic health records to genomics research, mainly focusing on women's health and the nuances of the sex differences in looking at genomics research. So let's get into it. Diseases that an individual presents changes over lifetime. This is a great representation from the study that was published a few years ago in Lancet. And this shows the frequency of about 300 most commonly occurring conditions that are diagnosed for an individual over lifetime. So we can see that how health changes over life, but so does diagnoses and testing and treatments. And electronic health records, or EHR, are a great resource because these contain wealth of information that is continuously populated on patients and is agnostic of any downstream research. EHR contains vast amount of data, especially patient data, such as family history, social history, and of course, medical history, which is collected at each encounter within the health system, as shown in this figure here. There are many components to this healthcare data. In general, EHR contains a wealth of longitudinal, real-world patient information that is collected in standard clinical practice. So this information includes demographic information on patients, medical and surgical histories, allergies and medications, diagnoses and procedures, along with any details that happens during the patient encounters. And this information is present on individuals both in terms of structured data in the EHR and also in terms of unstructured data, which is usually in terms of notes. Clinician notes. Now, with the increasing accessibility of genomic testing and a greater understanding of genomic variations, there are more efforts broadly to integrate genomics into this, into the healthcare data as well, or essentially more other omics data sets. So this is where my team, my lab comes into play. We look at these electronic health records and also genomic discoveries. We work on the point of looking at finding genomic discoveries and also looking at genomic discoveries that would be relevant to translational medicine. In my team, we have several projects that are ongoing, and in terms of linking the electronic health records with these genomic research, this is sort of the pipeline that we follow. So the first set here, you can see I'm listing some of the phenotypes that we currently work on. Many of these are in context of women's health in terms of doing deep phenotyping. Now, in terms of electronic health records, we are able to easily use just diagnosis codes to understand if an individual has a disease, for example, endometriosis, if they have a disease or they don't have a disease. However, many of the time, just the use of ICD codes or diagnosis codes that these are referred to as are not sufficient, because we do know many diseases are not equally represented in electronic health records in structured form, especially in diagnosis code, due to many measures. Many clinicians could represent a disease very differently, or insurance, because this is also based on how insurances deal with it. This may not also be represented in the electronic health record because of insurance. And also, if a patient is coming into a healthcare system way later than when they have a disease diagnosis, it is also possible that that disease would not be represented in a structured form, because that's not billable to a patient, or that's not a diagnosis they're getting at an encounter. Much of this information information is written in notes for individuals or in unstructured form. So we employ different methodologies, which I'm referring here to as deep phenotyping, to be able to extract individuals that have a disease and do not have a disease. Many times this is referred to as case and control type of studies. Then linking this to genomic research are the next two parts here, which are referred to as GWAS and XWAS. GWAS means genome-wide association studies, and XWAS mean exome-wide association studies. So genome-wide association studies are usually focused on looking at common variants that are in non-regulatory regions that are linked to these phenotypes, and we have some that are ongoing in my project, in my lab that are listed here, and some more. And XWAS, in the same way, are exome-wide association study, or focusing on the region of the exonic region of the genome. And here we're looking at more rare variants and how they are associated with these diseases. Both these measures are very useful in genomic discovery. The last part here is risk prediction. As I had said before, we take genomic discoveries that are useful for translation to then be able to predict individuals who are at a risk of presenting with the disease. So we have several projects that are ongoing in this context that are listed here as well. Specifically, as you can see, some of these projects like endometriosis, we do deep phenotyping, both genomic discovery in GWAS, XWAS, and also look at risk prediction because this is a more understudied phenotype. Whereas there are other phenotypes such as primary open-angle glaucoma, where a lot of genomic discovery has already been taken place, and we are looking at more risk prediction methodologies where we are taking these genomic discovery and trying to see how we can model this into being able to predict risk for individuals who present with glaucoma and who will not present with glaucoma. So the main question that we are really interested in understanding is what disease a patient will have. This is in context of risk prediction, or if we are taking in account in context of drugs or the effect of the drugs, we want to be able to predict how will a patient react to a drug. And there are many factors that are taken into account for understanding these questions, and these include demographic factors such as age, race, medical history, family history, genetic predisposition, as I had explained before, other drugs that this patient may take, and also their smoking and lifestyle. All this information that I'm listing here are accessible through the electronic health records, and we try to gather all this information together. Not only that we want to be able to predict what disease a patient will have at a given time, we want to be able to predict what disease a patient will have during the life course of their time, at different stages of their life. To do so, we use large biobank, specifically electronic health record linked biobanks that are useful for doing such research. One of such resource is Penn Medicine BioBank. Penn Medicine BioBank is a University of Pennsylvania health system linked biobank where any patient that comes into the UPHS health system is eligible to be recruited into the biobank. This is a single institutional research program, so it's only Penn Medicine and also all the hospitals that are linked with Penn Medicine. We do both in-person and remote recruitment for these patients and collect their blood via centralized processing. The blood is then used for extracting DNA, which is then sent out for genomic sequencing, and all this data on the genomic side is then linked to the electronic health record. So we do have information on these patients on how to understand how genomics link with their diseases. And also we do have permission to be able to recontact and do callback studies on these patients. This plot here is showing some demographic information about the Penn Medicine BioBank. We have approximately 275,000 participants that are recruited into the biobank. And also the biobank has exome sequenced and genotyped about 45,000 individuals. It is a fairly balanced biobank in terms of males and females. And also we do have individuals that are self-identified as white or European ancestry, and also a large number of people that are not self-identified as white, but also self-identified as other demographic populations. Another data set that we use is called eMERGE Network. It is Electronic Medical Records and Genomics Network. This is a multi-ancestry data set that includes about 100,000 participants across United States. This plot here shows all the different universities and healthcare systems that contribute data to the eMERGE network. Here we have on these 100,000 participants genotyped and imputed data available, and all the electronic health record that is linked with this genomic data. This includes not so much on notes, but mainly diagnoses codes, laboratory measurements, demographics, and vitals on these participants. Next, another large resource for doing such genomic research is the UK Biobank. This includes about 800,000 individuals that have their genomic variants and exome data linked with the electronic health records. And not only just the electronic health record, there's a large number of survey data also available on these participants. And in terms of electronic health records, it does include diagnosis code, but also imaging data that is available on these participants. And this is a biobank that is out of the UK. Last but not the least, another data resource that is very useful here is All of Us research program, which is a United States-based biobank. This has recruited about 400,000 participants so far, for which the electronic health record data is available, physical measures, some survey questions, even lifestyle data such as from Fitbit records. And then also there is whole genome sequencing data that's available on all of these participants. So I want to be able to focus on one specific project to give you an overview of some of the work that we've been doing in my lab, and that is in context of endometriosis. Our long-term, we want to be able to build risk models, especially integrative risk models for both early prediction of endometriosis and other associated long-term health outcomes. So why endometriosis is important? Over 200,000 women get diagnosed with endometriosis each year in the United States. And endometriosis is usually presents itself as by the symptoms of pain, as it is shown here. It's usually pelvic pain, menstrual cramps, and also heavy menstrual bleeding. However, the symptomatology of endometriosis is highly complex. As you can see on the right here, we see there are multiple symptoms that patients can present with that could relate to endometriosis, but every patient does not present with all of these symptoms. And not just that endometriosis is a disease of pelvis, it is now considered a chronic systemic disease, because endometriosis is linked with many other long-term outcomes, such as depression, anxiety, also cardiometabolic outcomes, and also cancers, such as ovarian cancer and also melanoma. So considering that, we want to be able to predict individuals who will present with endometriosis, but also who are these individuals that would present with long-term outcomes, because it's not inclusive that everyone who has endometriosis does not have all the long-term outcomes. We want to be able to use genomic information along with other risk factors for endometriosis to be able to predict that. And why this is important is why we want to do an early prediction, because right now the diagnosis for endometriosis is delayed by at least 3.6 years. Pain, as I said, is the main symptom of endometriosis, and pain is minimized in women heavily, and more so in women of color. So we want to be able to use information from genomic research to present this long-term goal of to be able to develop effective, precise, early screening tools to identify patients that are at risk for endometriosis and concomitant comorbidities. As I'd said before, there's so many symptoms of endometriosis, and why I emphasize here on concomitant comorbidities is because although endometriosis is diagnosed in over 200,000 women every year, 50 to 70% of those women actually come in for infertility treatments, and then they get diagnosed with endometriosis. So that's way further than when this could actually have been treated more. So we want to be able to use these models or develop the models to be able to predict endometriosis earlier. And lastly, we also want to be able to predict risk of associated long-term health outcomes, as I mentioned before. This plot here is showing some of the genomic research that has been done in endometriosis. Large genome-wide association studies have already been conducted on endometriosis that includes hundreds and thousands of individuals. And this plot on the left here is showing a genome-wide association study in endometriosis and its subtypes that are done in the study in 2023. This genome-wide association study identified about 42 different significant loci that are associated with endometriosis and its stages for endometriosis. And on the right here, you can see these 42 lead loci and what are the effect estimates for these loci in female only and female and male controls. And these effect estimates are useful in being able to use that for risk prediction. So this is great that we have 42 loci from genome-wide association studies. The next point is how we use this for any genomic risk prediction. To do that, we construct models such as polygenic risk score. Polygenic risk scores are weighted sum of effect estimates from genome-wide association studies summary stats to identify risk for an individual. As shown in the graph here, we take genome-wide association study on a discovery dataset. Based on those genome-wide association study signals, we identify the effect estimates, as I had shown in the previous slide, for all the variants that are associated or how they are associated with the disease of interest. Then we build a polygenic risk score per individual, which is essentially summing the effect estimates of all the SNPs that are significant for this association. And this could be just one SNP, 10 SNP, or hundreds and thousands of SNPs. When we are talking about only a targeted list of SNPs, then we refer to that as genetic risk score. Otherwise, we're referring to them as polygenic risk score or PGS, which is when we're including more than just a handful of the SNPs. Now, this helps us identify individuals on where they fall in this normal curve distribution, as shown in this slide, so that people that are on the left-hand side of this normal distribution are the individuals that are at low risk for a disease, and individuals that are on the right spectrum of this normal distribution curve would be at a high risk for the disease. So for the genome-wide association studies in endometriosis, we generated a polygenic risk score and then applied that polygenic risk score in our Penn Medicine BioBank population. We do this in a more stratified manner. We use individuals that are genetically identified as European descent individuals, and also individuals that are genetically identified as African descent individual, and see how this model predicts accuracy for these individuals. And we see that the area under the ROC curve for this model, which is polygenic risk score model, including age, principal components into the model, is about 0.6 or 60% accuracy, which is so far mediocre. It's not so great, because 50% accuracy would mean just flip of a coin, whereas 60% is a little bit better, but not so great. What we think the problem here is that, as I said, there are so many symptoms for endometriosis and also the diagnosis for endometriosis is delayed. We think that this is a problem of clinical heterogeneity. And this slide here explains a little bit more on what I mean by clinical heterogeneity here. I do not expect you to read everything that's written on the slide here, but to guide you through this, what we're looking at here in this box that is gray and blue and white is that the evaluation criteria that a clinician has to go through in terms of evaluating someone for endometriosis. So on the left-hand side, you see all the symptoms and the patient history that a clinician has to review to see if all the symptoms are consistent with endometriosis. Whereas on the right side in the gray, you see all similar symptoms and history questionnaires and symptoms that a clinician has to review to see if there is other diagnoses than endometriosis that may also be relevant, such as IBS or uterine fibroids or PCOS. So they have to evaluate symptoms that are consistent with endometriosis, but also evaluate symptoms that could mean something else. Along with that, a real diagnosis for endometriosis. For that, and a clinician has to go through a exploratory laparoscopic surgery to do an endometriosis diagnosis. Therefore, we want to see we want to test this hypothesis that since there are so many different symptoms of endometriosis, we want to see if this problem of not being able to correctly predict risk for an individual for endometriosis using their genomic information is due to the symptomatology that relates with the subtypes of endometriosis. So our hypothesis here is that endometriosis symptomatology and comorbidities that an individual present with may indicate possible subtypes. Okay, so the main objective of this study is to identify if the endometriosis subtypes using the clinical data and then understand if these subtypes relate to distinct genetic etiologies. To do that, we first extract clinical features from electronic health records. We extract about 39 clinical features that relates to symptomatology of endometriosis and comorbidities of endometriosis. So these are all the symptoms that I had listed before. If there's anything that has a ICD code or a diagnosis code related to these symptoms, then we extract them. We first perform some feature selection on these symptoms to then take that into a next step of clustering to identify subtypes of endometriosis. So we apply a hierarchical clustering on these feature-selected features of clinical risk factors that we extracted to then do clustering, and we were able to identify 5 different clusters of endometriosis. So as you can see from the plot here, which is mostly just showing the visualization of these clusters, the clusters are actually identified in a more data-driven way. And then we look back into the actual symptoms that patients has or comorbidities that a patient has that help us define these clusters more. So, and those are defined based on the prevalence of these symptoms. So what is the highest prevalence of different symptoms in each of these clusters? And we can see that we have 5 different clusters. Cluster 1 relates more to the pain comorbidities. So these are the individuals who complain more of pain and also have other comorbidities of pain, such as fibromyalgia. And then we have a second cluster that is uterine disorder. These are patients that have more other uterine comorbidities or uterine disorders. So they have either uterine endometriosis, such as adenomyosis, that's a subtype of endometriosis, or they have other complications such as uterine fibroids. These are the patients that fall in the second cluster. The third cluster we have are the patients that have pregnancy complications. So they have either higher rates of infertility or higher rates of C-sections or complications that happen during pregnancy. Then we also have another cluster, fourth, which is representing individuals with endometriosis that present long-term cardiometabolic comorbidities. So these are the individuals who have higher prevalence for hypertension or CAD or heart attack that is presented in the cluster 4. Then we have the last cluster, which is cluster 5, and it is the cluster we really don't know what is the most highest prevalence for. So we label this cluster as EHR asymptomatic. Now, I'm pretty sure if we look more deeper into these clusters, we're able to identify other things, but this is mainly a data-driven way that we approach this in identifying these different clusters to see how we can look them based on just the prevalence of the diseases that we have. Then we take this step further, as I've said before, that we have 42 loci that are identified for endometriosis. And most of those tests that are done for these 42 loci are in black and white case control status of endometriosis. Here, we take each of our subtype of clusters that are presented in the slide before, and we test the association of these 42 loci in these different subtypes. And the plot here is showing us, especially the bar charts here are showing us for each of these clusters, out of the 42 loci, which ones were more significant in each of these clusters. So the point of the plot here is to show that along with these endometriosis subtypes that are clinically heterogeneous and present different clinical risk factors in each of these subtypes, we also observe genetic heterogeneity. So, for example, clusters such as the cardiometabolic cluster only has one signal that was significant, and that is RNLS, which is a gene that is also already known to be associated with other cardiometabolic phenotypes. So we do see that endometriosis patients that also present with cardiometabolic phenotypes just have this one gene that is with different allele frequencies than other individuals. And similarly for uterine disorders, that is the cluster 2, we see high association of genes such as Wnt4, GREB1, GDAP1, and DNM3, which is not presented in many other clusters as well. So we see this sort of genetic heterogeneity that also links with the endometriosis subtypes. So that's interesting. Now, you must think that where are we going from there? Because I said that, that we want to be really able to use this information for doing more risk prediction. So we want to now take this into a step further where we would do larger genome-wide association studies for these subtypes and build polygenic risk scores that are specific to these subtypes, and then build integrated model that not only predict individuals at low or high risk for endometriosis, but also predict individuals that are very low risk that would not benefit from any sort of intervention versus individuals that may present with different symptoms, either pain symptoms or other symptoms that are related to endometriosis. So we want to be able to stratify our risk based on those symptoms. And for also individuals that are at high risk for things such as infertility or long-term outcomes, we want to be able to use this information that we learn from genetic studies to be able to predict how these individuals need interventions or what type of interventions they may need based on what risk profiles they fall in. I've given you a lot of overview on the type of the work that my lab does, and also getting deep dive into the endometriosis phenotype. I want to say that for many of these complex problems, they are not just easy solutions. Therefore, my lab focuses on the extraction of multimodal data from the electronic health records to answer many of these questions. We look at discovery studies, as some of which I had mentioned today, such as genome-wide association studies and exome-wide association studies, to do biomarker identification, doing clustering of individuals and disease enrichment. And then we take these a step further in doing some risk prediction to be able to identify individuals that are at high risk and low risk for a given disease, but more so also look at individuals that are at risk for long-term effects or subtypes of these diseases. With that, I would like to thank all the members of my lab and also all our collaborators, the Penn Medicine BioBank team that also is helpful and participates in many of these projects, and also the grant supports that I have received so far.